As you are studying for your cells and cell transport test, here are the things that you will want to make sure that you understand. So the first is the cell theory. The cell theory consists of 3 statements. Cells are the basic units of life. All cells are produced from other cells. All living things are made up of cells. When it comes to cells, there are 2 categories. The first being prokaryotes. These are very small and simple cells. They will never have a nucleus, although they do still have DNA. It's just not protected in a nucleus. It's just floating in the cytoplasm. An example of a prokaryote would be a very small, simple bacteria cell. And you can see an example of a prokaryote on the slide right now. You can see it is simple, doesn't have a lot of special fancy organelles on the inside of it. It can sometimes have cilia on the outside or a flagella, which is the tail-like structure you see on the back, which is used for movement. Eukaryotes are the other category of cells. These are much larger than prokaryotes. They're complex, they have a lot of different organelles in them, and they will always have a nucleus, which is the center of the cell, it's the control center, and the job of the nucleus is to protect the DNA for this eukaryotic organism. Plants and animals are both examples of eukaryotic cells. Those are the 2 that you should definitely know. The difference between plant cells and animals, you can look at the shape to tell the difference. So usually plant cells are going to be much more rectangular and rigid and angular. This is going to be due to that extra thick cell wall that's on the outside of them. That just gives them extra protection and strength and rigidity. Animal cells are typically going to be depicted as being more round and irregularly shaped. They do have a cell membrane on the outside, but they do not ever have a cell wall. Since they are both eukaryotic cells, they do both have a nucleus to protect their DNA. That is a key similarity that you need to know between them. Just they both will always have that nucleus. Some of the organelles that you definitely will need to know for your test. The nucleus, again, that's where the DNA is found in eukaryotic cells. Every cell has a cell membrane on the outside, which has the job of controlling what can enter and leave the cell. Cytoplasm is the gooey fluid that fills the cell. Every cell, no matter what it is, prokaryote or eukaryote, it has to have cytoplasm. Lysosomes are small round structures that do contain digestive enzymes to break down cell waste. Ribosomes are also small round structures, but they're going to be the smallest round structures, and these are the protein factories of the cell. Every single cell has to have ribosomes because they're making the proteins, and proteins just do so much for living things, for cells. They have to have ribosomes. It's a necessity. Mitochondria, the powerhouse of the cell, these organelles are going to make the ATP energy for the cell. It's the powerhouse, makes the energy. That's important. All eukaryotes are going to have mitochondria. Prokaryotes are not going to have mitochondria. These are very special, fancy organelles. Chloroplasts are also fancy and special, complex organelles. Chloroplasts are where photosynthesis happens, so they're going to absorb sunlight and use that to make a food called glucose. Plants have chloroplasts. Animals do not. So that is another thing you need to know that's going to differentiate plant and animal cells. So plant cells will have chloroplasts. Animal cells do not. A cell wall is going to provide additional support and protection. Plant cells have this. Animals do not. You also should know that prokaryotes can sometimes have this for protection. So prokaryotes, like bacteria, they do oftentimes have this extra layer on protection similar to a plant cell, but never animal cells. A central vacuole, that's another thing that plant cells will have. It's a very large storage organelle. It's got a lot of water in it, some nutrients, but it kind of just looks like a big bubble inside of the plant cell. Animal cells do not have a central vacuole. Here's an example of a plant cell. We have some important key organelles labeled here. You need to know these. So plant cells have a chloroplast. That's where photosynthesis happens. They have a nucleus because they're eukaryotes. That's where the DNA is found. They have a central vacuole, that very large storage bubble filled with a lot of water. Cell wall on the outside for extra protection. It makes it really stiff and rigid. Mitochondria, the arrow's a little bit off. That's pointing to the red oval shaped thing. That's where the energy is going to be getting created. And then the cell membrane lies just inside of the cell wall. And again, cell membrane is on every single cell, no matter the type, and that just controls what can go in and what can leave the cell. These are the things you for sure need to know about a plant cell. For an animal cell, definitely know it's a eukaryote. It's got a nucleus in it that protects the DNA. Since it's a cell, it's got to have a cell membrane, and it has to have the mitochondria to produce the energy. It does have other organelles, but I would say these are the key things you'll need to know for your test. You also for sure need to know these things. I've already mentioned this, but just to reiterate this here, all cells, no matter if it's a prokaryote or a eukaryote, must have a cell membrane, DNA, cytoplasm, and ribosomes. For the cell membrane, there are additional details you need to know specifically about this organelle. It's made out of 2 rows of phospholipids that look like the little jellyfish. We sometimes call a cell membrane a phospholipid bilayer. It controls what can enter and leave a cell. It's very picky. We say it is semi-permeable or selectively permeable. Some things can enter and leave, others are not permitted to do so. Inside of the cell membrane, you'll often see transport proteins. So the channel protein, like the one you see here, is like a tunnel. Some molecules, when they're going through the cell membrane, can't go straight through the phospholipids, and they must utilize these transport proteins. The molecules that can go straight through the phospholipids can utilize the type of transport called simple diffusion. This is the first type of passive transport you must know. These molecules are going down their concentration gradient from a high to a low concentration, straight through the phospholipids because they're small and they can fit all on their own. The other type of passive transport you need to know is facilitated diffusion. These molecules are still going from high to low, but they have to go through a special transport protein to get to the other side. Passive transport does not use any energy. It's easy for a cell to do this. It just is happening all on its own. Unlike active transport, which is when a cell is using energy to force these molecules to go against their concentration gradient. These molecules are going from a low concentration to a high concentration through a carrier protein, sometimes referred to as a protein pump. ATP energy must be used to do this. The final type of cell transport you need to understand is osmosis. This is a type of diffusion, which is passive transport, but it's specifically the movement of water molecules, which is H2O. The terms you need to know very well for osmosis are hypertonic, hypotonic, and isotonic. Hypertonic means more solute. Hypotonic means less solute. Isotonic means the concentrations of solute are equal. Hypertonic and hypotonic are actually opposite terms. Where you find one, you will find the other. So if you're told one area is hypertonic, then by default, the other area must be hypotonic. Water will always move to the area that is hypertonic, the space with more stuff. So in this diagram, the water is going to move to the right because that's where there's more solute. It's trying to dilute that higher concentration of stuff. What happens if a cell is placed in a hypertonic solution? The water is going to leave the cell, and the cell is going to shrivel up. What happens if a cell is placed in a hypotonic solution? The water is going to go into the cell, and the cell is going to swell. These are the things that you need to know for your test. If you have any questions about this, any of this stuff, please let me know. Happy studying!